Hey guys, welcome back. This is MCAT Day 6 continued. I need to do I need to do one more hour, so let's do it. Oh, okay, go for it. By the way, when that's on, I think the mic picks up pretty good.
What?
No, I forget. All right. Okay, let's hit the timer on the clock. Well, not yet. Let's get some gloves.
Eat up.
That's right. I gotta eat up. I got my milk. Milk contains that protein. Makes you a little bit sleepy. A little bit sleepy.
Oh.
So that's why I got the milk.
Then no game, man. Maybe you'll get a game.
That's a good game. I can take like 3 hours for sleep. Open. Open.
Open what? My organs?
Open.
C-section.
Can you stop?
I'm sorry.
All right, guys, let's get to it. Oh my god, my shoulder's kind of huge. Look at that shoulder. Got a good shoulder on me.
Nice.
I'm kidding. Okay, let's get started. This one seems pretty good. That's not true. That's not true. I think it's A. Let's go.
Wait. May 7th is Jonah's birthday.
Oh. I'll be here.
June 10th, David. Oh,
Oh, okay.
what's this?
Big boss. Big boss. Big boss, UCI. So when glucose is low and lactose is abundant, the upper arm will turn on. Oh, cool. This makes sense. Oh my god.
Makes me feel like this.
Why are you on there? Why are you not on there? Who's that? Ash Wilson. Oh. Opposite. No, it won't even get. It won't even transcribe the genes right on my microphone. It's not B, it's not C. Maybe A, but I don't think so. I'm gonna say D. Oh my god. I'm getting them all right. Am I so good? I mean, I would think that when it's methylated. Mm. Oh, deacetylation. Well, I think this would loosen it. This is oh man. Oh, this is D amp or decamp or whatever. Decamp. So that means that this is gonna tighten the structure, be a heterochromatin, and not allow access because it tightens it. But not uncoiling. This one. Yeah. Methylation. Maybe this one. 5 prime is the 5 prime is a methylated guanine. I'm gonna see if it's usually used for protecting it. I don't know. I guess that's the poly A tail. Um, Maybe if it's not a protection, it's promotion. Oh man, polyadenylation. Polyaden, ah, polyadenylation. Polyadenylation. Maybe this one. Oh man, what does that mean? Oh, the question is which one the phone does not. You gotta read the question. It's gene silencing. Professor Biden. That doesn't make any sense. A polypoprotein of macrophages. Woohoo! 73, at least that's better, no? Something. Oh. I added these today. Actually, I think I added translation. Maybe not. I will just quickly watch these and just see what happens. Oh man, we just have to read it and then we'll watch them. Tomorrow we'll be reading the yield guide, and then we'll do medallion. DNA Tech. Hey Chucky, how's it going? How's it going, brother? Hope you're doing good. I gotta fix this camera. Like, it's a different one, but I'm out here. Studying to take the MCAT. I'll make a video about it. Maybe maybe I don't know if I'll make it tomorrow, but I'll make a video. Yeah, I know. I think just for the weekends, I'm gonna do this angle. I think just so maybe people get more involved. Yeah, I know. Just my back. I know. My back. Yeah, I'll do a side view Mondays through Friday, and then Saturday, Sunday. Oh, sorry. I'll do Friday to Sunday a side. Friday to Sunday a back view. Monday to Thursday a side view. Yeah. All right, brother. I gotta get back now, but I mean, thanks for being here. You're one of the OGs of the channel. Yeah, I still use the gloves. They're right there. I was just eating a quesadilla. All right, brother. I got it. I gotta hit this. Oh, you guys aren't even seeing what I'm doing. Damn. Man, Chucky, thanks for being here, though. Glad you're here. Okay. What do you guys think about this? Let me go back. Okay. Okay. AUG. Wait, not AUG. Oh, UAG. UAG. 'The reading frame is not shifted. Oh, I guess so. very centric, very paracentric. Oh my God, you know. Oh, I should probably put it in a new chat. Oh, 92 chromosomes total. "46 chromosomes total." Let me see. There's a fusion of two central chromosomes into a single chromosome. Down syndrome. It's a chromosome. New activity. New activity. Activity. G so regular sequence. Adding between test 123 and 24. The repeat plus 123 and 126. G524B presents a message in return. Okay, so oh, oh, for the protein is italicized. Gotcha. Mutation in the CFTR gene can result in a mutated CFTR protein. CFTR gene is italicized, CFTR protein is not. Right. Both copies of the CFTR gene are inactive. Oh.
Into genetic mutations. But first, let's go over the central dogma of molecular biology, which is just the idea that genetic information is... Today, I'm going to give you a quick introduction into genetic mutations. But first, let's go over the central dogma of molecular biology, which is just the idea that genetic information in a cell is stored in the form of DNA. And this DNA is used to generate complementary RNA through a process called transcription. That RNA is then used to synthesize a corresponding protein through the process of translation. So looking at a quick example, our short DNA strand here will be used to generate an RNA strand. Remember that A pairs with U, protein, through the process of translation. So looking at a quick example, our short DNA strand here will be used to generate an RNA strand.
Okay.
Remember that A pairs with U, or T, E, and C pairs with G. Remember that A pairs with U, or T, and C pairs with G. Strand here will be used to generate an RNA strand. Remember that A pairs with U or T, and C pairs with G. Next, our RNA will be used to generate protein through translation. And remember that during this process, RNA nucleotides are read in groups of 3 called codons in order to generate corresponding amino acids. Now, just very generally, we say that mutations have the effect of making this synthesized protein not turn out quite right. So I'm going to give a quick shout out to sickle cell disease, which is an example of a disease that's caused by a genetic mutation. So you may remember that there is a protein in red blood cells called hemoglobin, which we can also call HB. And hemoglobin is a protein that coordinates to iron ions in order to hold on to oxygen molecules and transport them throughout the body. Now, the mutation that causes sickle cell disease results in a mutated form of hemoglobin called HbS being formed, where the S is for the word sickle. And the difference between normal hemoglobin and HbS is that one glutamate amino acid residue is being replaced with a valine amino acid residue. And this small change results in all of these mutated HbS proteins aggregating together in a red blood cell, which makes it very difficult for that red blood cell to transport oxygen effectively. Now, just a side point, remember that red blood cells are initially generated from hematopoietic stem cells through a process called hematopoiesis. So where are mutations found, and how did they come up in the first place? Well, let's look at a couple different possible mistakes that could lead to an incorrectly produced protein. So first, we'll see what happens if a cell makes a mistake during translation, and we'll stick with our example of sickle cell disease from before. So let's say that we have this sample piece of DNA with 3 nucleotides from the gene coding for hemoglobin. This DNA is transcribed to form the complementary RNA sequence GAG. Look at a couple different possible mistakes that could lead to an incorrectly produced protein. So first, we'll see what happens if a cell makes a mistake during translation, and we'll stick with our example of sickle cell disease from before. So let's say that we have this sample piece of DNA with 3 nucleotides from the gene coding for hemoglobin. This DNA is transcribed to form the complementary RNA sequence GAG. Now that GAG would normally correspond to a glutamate residue during translation,
Let me read this.
But a mistake during translation might lead to a valine residue being translated instead to produce the mutated hemoglobin associated with sickle cell disease. But notice that if a mutation happens during translation, the cell will only produce one mutated hemoglobin, or HbS, for each overall mistake, encoding for hemoglobin. This DNA is transcribed to form the complementary RNA sequence GAG. Now that GAG would normally correspond to a glutamate residue during translation, but a mistake during translation might lead to a valine residue being translated instead to produce the mutated hemoglobin associated with sickle cell disease. But notice that if a mutation happens during translation, the cell will only produce one mutated hemoglobin, or HbS, for each overall mistake. And since cells are making tons and tons of hemoglobin, just one mutated protein might not have that big of an effect on the cell. So we can say that mistakes during translation probably don't cause mutations like the one associated with sickle cell disease. So next, we'll look at mistakes during transcription. Again, we have our CTC piece of DNA, which would normally make GAG on RNA, but maybe a mistake occurs which leads to the transcription of a GUG instead, which would then code for the valine associated with mutated hemoglobin. Now, if this mistake occurred, the cell would only make a few mutated hemoglobins for each mistake, since an individual strand of messenger RNA will only be translated a couple of times before being degraded. So we can say that mistakes during transcription probably don't cause mutations like the one associated with sickle cell disease. Finally, we'll look at mistakes in the DNA strand. If our CTC in DNA is mistakenly turned into a CAC, then our corresponding RNA from transcription will be changed, and ultimately a valine would be produced instead of a glutamic acid. Now, since a cell's DNA stores all of its genetic information, that mistake would lead to all future hemoglobins produced from that gene being mutated. So overall, we can say that mutations will usually result from mistakes in a cell's DNA, and not from the RNA or the protein. So where do these types of mutations come from? Well, there are two ways a person can get a genetic mutation. The first is that they inherit it from their parents. Remember that DNA is passed down from parents to offspring. So if we have a mutated father here, then there's a good chance that at least one of his kids will inherit that mutated gene the same way that the child might inherit any amount of that parent's DNA. The other possibility is that the mutation will come on spontaneously, which is where a person suddenly gets a mutation in their DNA without their parents having had the same mutation. And spontaneous mutations can come from many different sources, with just a few examples being from DNA replication errors, environmental factors like certain poisons, or it's also possible that genetic mutations can come on entirely randomly. So what did we learn? Well, first we learned that mutations originate at the DNA level and not at the RNA or protein level. But the effects of a mutation, like the example we gave with sickle cell disease, are found with problems with the proteins that are ultimately expressed by the mutated DNA. Now, like every rule, there are a couple of exceptions to this one, but we can say that the effects of a mutation are usually found at the protein level. And finally, we learned that mutations are either inherited from a parent or come on entirely spontaneously. So today we're going to talk about the different types of genetic mutations that you'd find in a cell. But first, I want to review the central dogma of molecular biology and how the genetic information of a cell is stored in the form of DNA, which is then transcribed to form RNA and then translated to generate protein. Nucleotides from the DNA are transcribed to their complementary forms on RNA, which are then read as codons, or groups of 3, to code for specific amino acids in a larger protein. Now, if you mutate one of the nucleotides on DNA, like let's say turning this thymine base into an adenine base, then that will affect the RNA sequence and ultimately the protein that follows. So we say that mutations are mistakes in a cell's DNA that ultimately lead to abnormal protein production. So what are the different types of mutations? Well, the first type of mutations we're going to talk about are called point mutations. Now here I've just written out a random sequence of DNA, which is just a repeating pattern of CTC, which would code for a repeating sequence of GAG in the RNA strand, and finally a protein sequence of 3 glutamate amino acids. So a point mutation is when one of our DNA bases is replaced with another. So in this example, a thymine base is being replaced with an adenine base, which leads to a change in one RNA nucleotide and ultimately a change in one amino acid. Another type of mutation is called frame shift, which works a little differently. So first, I'll write out the same DNA, RNA, and protein sequences from before, but now instead of changing one base to another, I'm going to add one to the sequence. And here I've thrown in this extra cytosine base that I've written in blue. Now, naturally, this change would lead to an additional guanine base being in the resulting messenger RNA sequence. But what's interesting is that this mutation will change the reading frame of the RNA. Remember that RNA is read in groups of 3, or codons, when being translated to form protein. But now, since we've added an extra G here, all of the codons coming after that extra G will look a little differently. Now, instead of having 3 GAG codons, we've swapped out 2 for GGA codons. This means that 2 of our amino acids in the final protein will be changed. And in this example, they'll be changed from glutamate to glycine. So you can see that frame shift mutations usually have more significant effects on the final protein than point mutations do. Now, it's important to recognize that both of these mutations are classified and named for how they affect the cell's DNA structure, and aren't really named for how they affect the resulting protein. Now, our next type of mutations are nonsense mutations and missense mutations. Let's say we have a DNA sequence that normally generates RNA and codes for a cysteine amino acid. A nonsense mutation is any genetic mutation that leads to the RNA sequence becoming a stop codon instead. Now, missense mutations are a little different, and they're any genetic mutation that changes an amino acid from one to another. So in this example, our mutation is changing the resulting amino acid from a cysteine to a tryptophan. Now you can see that nonsense mutations probably affect the resulting protein a lot more than missense mutations do, since that new stop codon that we're creating could chop off a huge section of the protein instead of just changing one amino acid to another. So now we can divide the missense mutations even further into a bunch of smaller categories. Silent mutations are when the mutation doesn't actually affect the protein at all. Since many different RNA codons can code for the same amino acid, it's possible that the mutation might not affect the protein at all. So in this example, CCA, CCG, CCT, and CCC in a section of DNA will all end up coding for glycine. So if you change the third base, it wouldn't affect the final protein. Conservative mutations are where the new amino acid is of the same type as the original. So here I have a glutamate and an aspartate, which are both acidic amino acids. So a mutation that swapped out an aspartate for a glutamate would be a conservative mutation. Finally, a non-conservative mutation is one where the new amino acid is of a different type from the original. So here we have a serine amino acid, which is a small polar amino acid, being replaced with phenylalanine, which is a large non-polar aromatic amino acid. And this would be an example of a non-conservative mutation, since serine and phenylalanine are different types of amino acids. Now I'll point out again that all of these mutations are classified and named for how they affect the resulting proteins, and aren't really named for how they affect the cell's DNA. So let's look at a quick example. Sickle cell disease is a disorder where hemoglobin, or HB, which is a protein found in human blood, is mutated into a less active form, which we're going to call HBS. And it results from a single glutamate residue being converted into a valine residue. Now we can classify this mutation as a point mutation, since only one DNA base is affected, but we have for valine and polar level. And second, we learn their effects on and then we have mismutations.
All right, guys, I'm gonna just end it here. Hey, let's go. Oh, we got some people in the chat. I just see you back. You're a great doctor. Hey, thank you, Famadine. I appreciate that. Hey, K-pop. Hey, good luck, man. I took the MCAT back in 2022. I'm studying for step 1. Thanks. Thanks, M to the lead. Thank you, guys. Thank you so much. Thank you, guys. Thank you so much. Very kind. I am going to sleep. You world. Yeah, right now, I have to do content review. I'm just doing. I'm following this plan here, which my friend, very close friend, used, and he got into medical school. So, well, I mean, not necessarily that means I'll make it, but being able to practice and read those explanations very thoroughly. Yeah, I know. I am actually really far behind. I won't have time to do the Anki today. I'm too tired, but this is one lesson I added today, and then I did. Oh man, I did. How many lessons? I think I did 3 lessons today. I did 3, well, almost 4, basically. So I have to add all these. We'll have a big Anki review day tomorrow for sure. No, I haven't taken any yet. Not yet. I at least want to take it. Well, I'm gonna take it week 4. I'm trying to follow this as best I can, really. Week 4, week 8, week 12, and then after week 12, I'm just gonna bring out a bunch of practice problems and New World and everything like that. You know, how long until the exam? Exam is July 25th, but I might push it back, seeing as I'm going a bit slow, to be honest. Yeah, I might push it back. Yeah. This is all very new to me, to be honest. Like, well, I mean, this concept of studying, and then there's just quite a lot that I that I need to remember. You think so? Oh, you think so? Hey, let's go. Thanks, Chidli. Chidli. Okay, yeah, hope so. Hey, man, that's oh, that makes me feel great. We'll see. I don't know. There's quite a lot of Anki review for tomorrow, and then I have to read the high yield guide. Where's the high yield guide? Where'd I put it? I have to do this tomorrow. I have it all mapped out though, so tomorrow will be a great day. Today was a great day, to be honest. I think tomorrow will be an even better day. Um, Hopefully, I don't know if I'll be able to wrap everything up tomorrow, but I feel like maybe I'll be like a day or two behind, and maybe one day I can like in the days coming up, right? Maybe on week 2, I can try to be on track, so or get ahead, or we'll see. Some subjects are, I think, easier than others from what I still remember. Um, so we'll see. I don't know. We'll just see. But yeah, I'm excited. I'm very excited. Very, very excited. Which deck? I'm not sure, to be honest. My friend set it up for me. Um, this is how it looks like. Uh, MCAT physics. There's like a whole lot in here. I think Penkow. I heard a lot when I'm on the MCAT subreddit. I don't know. I maybe shouldn't be on there, but anyways, yeah. So I hear a lot. UWorld, Pancow. Um, I am going to do all these full links though. Take it seriously. Wake up probably earlier than I am now, and then take it 8 hours just as if I was taking the actual exam each time. And then probably I'm thinking of taking a day after each full length to review it. And then just continue to follow along this strategy. Yeah. That's my strategy. Maybe I'll make a video about it. So yeah, that's where I'm at. I don't know if this will help anybody at the end. We'll see what my score is. I don't know, but I don't know. They're the gold standard. You should hold off. You should hold off on your AAC exams until the end. They're the gold standard. Okay. Yeah. I mean, I think one of the problems would probably be that I might memorize the questions, but I don't know. I just still feel like I'm so behind. So I think I'll just review it. Maybe. I don't know. Use the blueprint and Kaplan foldings first. Everyone feels behind. Yeah. Yeah, I feel very behind. Well, thanks for the advice, though, Actually, I appreciate it. I mean, yeah, there's a lot. I was thinking of going through this, as because it's already built in. Like this is built into the curriculum that I'm following with this. I was thinking, like I said, of taking the practice exam 1 at the very end to simulate the real exam and just kind of see what my actual score would be. Yeah, but thank you. I appreciate it. Thank you for the advice. Yeah, I appreciate everything. All right, tomorrow will be a big day. So that's all for me for now, guys. Thank you so much. Have a good night. I'm super tired.